So here we're looking at biomass, which is the focus of chapter 4, and that's this section of the carbon cycle. And sponge production of biomass is another way that sponges could be storing the carbon from the DOC and preventing it from escaping the reef. And many species grow quite large, so sponge biomass probably represents a significant quantity of carbon on coral reefs. So this is a reasonable alternate explanation to them producing lots of detritus. Since sponges are accumulating this biomass, it's important to know how fast this is happening, which means you need to know some pretty simple things about them, namely their growth rate, or how fast they change volume over time. But this isn't really easy to measure for sponges. For one, it's hard to estimate their age because they don't have any indicators of annual growth, so things like tree rings. They also don't have, they grow really sporadically and have irregular shapes, so this makes it difficult to measure their size. Fortunately, sponges don't move, and so for any structure that has a well-documented history, like a shipwreck, we can pinpoint the oldest age of the individual growing on that wreck if we assume that it could not have recruited to the wreck until it sank. So our objectives for this chapter were to follow this logic and measure the growth rates of sponges growing on the Tibbet shipwreck in Cayman Brac. And this was the first project that I worked on here at UNCW, and we published this in 2019. So here's the location of the shipwreck in the Caribbean. It's on the northwest side of Cayman Brac. So the Tibbet is a 300-foot Russian ship that was built in the 80s and purchased by the Cayman Islands government in the 90s and scuttled off the shore of Cayman Brac in 1996. It's the only diveable Soviet shipwreck in the Western Hemisphere, and it now hosts a thriving benthic community as well as tons of reef fish. So it's a really popular dive site. This wreck was sunk in 1996, and we measured these sponges in 2017. So we started with the assumption that their maximum age was 20 years. But then I was on YouTube one day watching footage of the wreck like this, and there's tons of these videos through the 2000s and 2010s, thanks to the popularity of this wreck and also the increasing availability of underwater cameras, things like GoPros. So what I did was find a lot of this footage from the last 20 years, figured out when it was filmed, and then I put together this timeline of images showing two parts of the wreck that were heavily photographed. And this showed something pretty interesting. So before 2006, the surfaces of the wreck were really bare. There was nothing growing on them. But then from 2010 on, there looked to be this very lively, healthy benthic community, including lots of sponges. And when I looked into reasons for why there might have been this delay in benthic community development, I learned that there were several hurricanes that impacted this region between 2002 and 2008. And a couple of these were really severe storms. So Hurricane Ivan in 2004 actually tore the wreck in half. And then the latest storm was Paloma. This was a Category 4 storm that hit the Cayman Islands directly. And here is a picture of the eye wall of Paloma going right over the Tibbets. So it's reasonable to assume that any sponges that had recruited during this period were probably swept away by storms, at least until November 8th, 2008, when Paloma cleared the island. And then after this, there were no major storms through 2017. So between November 8, 2008, and when we measured these sponges in August 2017, this came out to 8.74 years. And this is the amended value that we used as the maximum growth rate, the maximum age, sorry. And then it's just a matter of measuring sponge size, which, like I've said, is traditionally pretty hard to do for sponges because to measure their volume, you have to pretty much collect them and then measure it by displacement, which is extremely destructive and kills whatever you're collecting. But now we have more sophisticated tools like photogrammetry that we can use to accurately measure the volume of these individuals without even having to touch them. And all this requires is a camera and a scale bar. So how this works is we went out into the field and took a bunch of pictures of these sponges that were on the wreck. And then we used a special photogrammetry software called Metashape to align these photos and make a point cloud that we then did some more processing to and made a dense point cloud from it. And then eventually solidified that point cloud into a mesh and then made that mesh into a fully textured model that we could measure the volume from. And we measured the volume from these 3D models, and we did this for 38 individuals from these 16 species. And then we calculated a specific growth rate for the largest individual of each species, assuming that the largest individual is the oldest. And then we combine these specific growth rates with ones that were previously calculated in one of Joe's earlier papers, where they measured the displacement values and estimated the growth rates of sponges growing on the Spiegel Grove shipwreck in the Florida Keys. And combining these two data sets, we were able to estimate the growth rates of these two functional groups, so the chemically defended and palatable species. And here what we found was this general division where the defended species tended to grow a little bit slower than the palatable ones. This was pretty cool because it agreed with this idea of a growth defense trade-off, which reflects the two strategies that sponges can use to deal with being grazed by predators. So basically they can expend their limited energy towards producing chemicals that prevent them from being grazed, or they can expend that limited energy to grow really fast and compensate for being grazed. And also in the context of this research, if palatable species do grow faster and make more biomass, then they are moving more of the stuff up the food chain and turning it into a food source for more reef organisms. And therefore, moving it up the food chain faster than their chemically defended counterparts. And then just for an update, 5 years after we collected those measurements, on a whim the other day, I went looking for more recent videos of the tibbets, and I found some great ones, including this nice footage of a hawksbill turtle and 2 angelfish just chowing down on this geodia. And this was the only palatable species that we had found on the tibbets when we measured these sponges. So it's pretty cool to see this kind of grazing pressure in action and see why these species might have to grow faster to deal with this. And then I just want to give one more update of what we are continuing to do with photogrammetry. This is actually one of those, I think, COVID side projects that I think a lot of us had. And I got to work on this with a former undergrad and now alum, Eric Plessett. So we had these 3D models of giant barrel sponges that, or Gestispongia muta, that we had made as part of the Florida Keys monitoring of the species. And some of these were healthy and some had been sheared away, likely by the 2017 hurricanes that had impacted that region. And the cool thing about sponges is that they are really good at regenerating and regrowing, even if just a thin layer of their body is left on the substrate after their injury. And so you can see that kind of recovery there on the right. And these recovering and healthy individuals grow really differently. We've just never really been able to quantify that. And so we had these 3D models for 2018 and 2019 for each of these groups. And so here we're looking at the 2018 model of a recovering individual. And then we lined up the 2019 model over it using the special alignment method. And you can see that superimposed over it. And then we subtracted the distance between these 2 meshes. And here's what we got as a result. So we have areas in red where the sponge grew and areas in blue where it shrank. And when we compared the 2 groups, we found some pretty interesting differences. For one, the growth of the recovery muta was really concentrated around their oscula. And so they presumably need to regrow this oscula in order to have a functioning aquiferous system, to be able to pump seawater through their bodies. So this is one of the few essential parts of the sponge body. Meanwhile, the growth of the healthy individuals was more dispersed around their body, which would maximize their gain in volume and surface area, and also likely their feeding efficiency. So there seems to be some kind of trade-off here and reallocation of energy towards recovering after a severe injury versus just business as usual growth to optimize your feeding abilities. But that was just another project that we have in the works, and I thought fit in nicely with the other photogrammetry stuff that we did. And now back to this focus on the DOM, detritus, and biomass. So even though this research is so broad in the questions that we're asking, I think that it's really ties nicely together and tells an interesting story and also gives an important degree of nuance to this claim that sponges are important for cycling carbon. For example, in chapter 1, we found that only HMAs took up DOM. And if the sponge loop hypothesis is true, then these species should be producing a lot of detritus or growing fast and making a lot of biomass. But so far, we have found little signature of sponges in the detritus on the reef. And also, HMAs tend to grow slower. And this is because a lot of the species that are HMA also tend to be chemically defended. This is a figure from a recent review that showed that the HMA species tend to be less palatable because they produce defenses, while the LMAs are more palatable because they don't. And then just adding the trends that we saw in this research, so the differences in the DOM uptake and growth rate from chapters 1 and 4. We also know from previous studies that LMA species take up more particulate organic matter, or POM, than HMAs. So it seems like the whole diversity of sponge species is important here for accessing different fractions of the carbon, ranging from DOM to POM, and then bringing that back to the reef, and then either moving it up the food chain by being grazed, or sequestering it in chemically defended biomass. Basically, sponge species cycle carbon differently, and many species probably don't even complete the sponge loop as it was originally hypothesized, at least for the detritus aspect. But I'm not sure that that's such a bad thing, since detritus may not be limiting on these reefs to begin with. And given everything that we know about sponges, about their ability, all the places they can live, all the things that they can eat, and their ability to survive and hold space even after really strong storms like this recovering barrel sponge, I think that any benefits that sponges might add are probably outweighed by the problems that they're causing on these reefs, especially related to their ability to preempt space and outcompete the corals that are remaining on modern day reefs that are just so stressed out from other anthropogenic stressors. So that was the downer half of the conclusion. But on the bright side, I'm encouraged by all the things that are becoming available to us to study these systems. So even here, we combine traditional field and lab-based techniques with things like photogrammetry, metabolomics, even crowdsourcing data from YouTube. And I think that these and other techniques, like machine learning especially, will help us disentangle these complex interactions among these benthic organisms. And I'm excited to see where this research is going, and I hope to be a part of it as well. In the short term, I'll be finishing up here at UNCW in the beginning of 2023, working on publishing chapters 2 and 3 and the time series photogrammetry manuscript, and then helping to get the next phase of the DOM project off the ground with some new experiments. And then I'm excited to move back to the Virgin Islands for my next adventure, working as a postdoc at UVI on this Ridge to Reef project with Dr. Tyler Smith and other PIs and collaborators. So it's certainly been a roller coaster of almost 5 years, but luckily I've had so much help and support along the way. So first I have to thank Joe and my committee, Wendy, Ralph, Patrick, Nikki, for teaching me so much and pushing me out of my comfort zone. By out of my comfort zone, I usually mean into a chemistry lab, which is not my natural habitat by any means, either the high-tech one at UNCW or the not so high-tech one at Caribou. But really, it's been an excellent experience, and I'm really happy that I got to work with amazing people who help so much in the field, especially. So thanks to Steve, Laura, Alec, Chris, Michael, Melissa, Jim, and Sasha, as well as the staff at Caribou and the Aquarius Reef Base. I also have many people to thank for teaching me how to do some very necessary lab work. So thanks to Chad and Kim and the whole Disco Lab. I see many of you guys out there. Thank you all. And then I want to thank my former DIS students, Eric and Evan, who assisted me and pulled some long hours on these projects. And I know that this work would not have gotten done without them. Then on to the people that kept me sane. I have my awesome friends that I always have a great time with. They're from UNCW, from UVI, and even from Miami. Some of them are watching now, I hope. And I'm really grateful that they're willing to take 45 minutes out of their day to hear me talk about sponges. And of course, my family and Sam, thanks to my parents, first and foremost, who I think are watching. I couldn't have done any of this without their support and encouraging my interest since day one. And then Sam, who is here, who's just been so supportive and understanding and a great cook. And there's no one I'd rather run from hurricanes or hide from pandemics with. And I'm so lucky to have him and them and all these amazing people and my cat, Michael. Finally, thank you all for listening and attending. And now I'm happy to take your questions.